Thank you so much, Paige, for the nomination and for the opportunity to talk to you folks about some of the work that's going on in my lab. Some of you know that my lab really studies two different things. We study drug addiction and sort of epigenetic mechanisms involved in plasticity associated with addiction behavior. And then the other half of the lab studies neurodevelopment with a really strong emphasis on the topic that I'll be sharing with you today. So as you can see, the title of my talk takes MEF2 to tango. This is a story that's been ongoing since I was a postdoc in Mike Greenberg's lab a long time ago. And Jay Bykoff, who's on the call, was a lab member back then. So we have a shared history. It's always fun to talk about this. We just had a paper in biological psychiatry, came out a couple of months ago. And it's really been the culmination of 15 plus years of different parts of the story. So I'm going to kind of walk you through, whoops, see if I can figure out how to advance the slides. So, you know, during early brain development, it's really a complex process of neurons extending axons and forming their connections, the synaptic connections, with appropriate partners. And that process is a little messy. Axons, you know, have to follow cues and try to get to the general place where they need to form appropriate connections. And then they begin a dynamic process of forming synaptic connections, which is really massively occurring in humans in early prenatal and then early postnatal periods. And in mice, a lot of this is happening, again, you know, several days before birth. But really a lot of the synaptogenesis part, the formation of these synapses, is happening in early postnatal life. For most parts of the brain, the synaptic connections that form initially are thought to be fairly crude. And the brain takes signals from patterns of activity, and it refines the synaptic connections so that the final mature neural circuit functions appropriately to allow us to behave and function in our environment and be responsive and plastic to changes in our environment. There are two major types of ionotropic synaptic connections in the brain, those that are excitatory, driven predominantly by glutamate, the neurotransmitter glutamate, and the inhibitory synaptic connections, which are largely GABA mediated. Of course, they're neuromodulators and a lot of complexity, but I won't get into all of that. I realize a big chunk of the audience is probably not neuroscientists, and I don't want to get in too much into the weeds, but it unfortunately has to be a little bit. So, you know, the thinking very simplistically is that as the brain is maturing and forming, you're getting this balance of inhibitory and excitatory connections, and the dynamics of that need to be well balanced to allow the brain to function properly. And if you shift that balance by inappropriately forming too many excitatory or too few excitatory or too many or too few inhibitory, that can shift the balance, and that can tip to neuropsychiatric illnesses. So for example, epilepsy, seizures may be due to too much excitatory transmission and dysregulation of the EI balance. And one very popular, but again, overly simplistic view of one of the underlying causes of autism is that there may be a shift in this EI balance. And perhaps for individuals who show sensory hypersensitivity and some of those types of phenotypes, that may be, or seizures, this may be caused by a preponderance of excitatory connections versus the inhibitory feedback process. And while I don't put a lot of stake into this general idea, I think it is a useful construct for most of us to think about what may be going on at the level of a dysfunctional brain for a number of neuropsychiatric illnesses, particularly those on the developmental side of things. Oops. So this led me a number of years ago to, many years ago now, to study a family of genes called MEF2 transcription factors. There are 4 of these in the mammalian brain, and they are highly conserved, as you'll see again later. They function by having a very highly conserved DNA and binding and dimerization domain on the left-hand part of your panel. I don't know if you can see can you see my arrow moving?
Yes.
Yes, okay. So, oh good, well that's helpful. So the MADS and MEF domain control DNA binding and dimerization And one of the things that MEF2 transcription factors do is they form homo and heteromeric dimers, and they bind to parts of the genomic DNA that have an appropriate DNA sequence, which we call the MRE, or the MEF2 response element, which is defined by an AT-rich region. And MEF2 then binds to a number of numerous targets in the genome and regulates gene transcription. And one of the things that's interesting about MEF2 transcription factors is that they're highly regulated by neuronal synaptic activity. So when a neuron is very active, enough to depolarize the neurons, this turns on programs of gene expression that can lead to changes in synaptic connectivity, or the elimination of synapses, which is what I'll tell you a little bit more about today. So some of the early evidence for this came from a paper based on work that one of my colleagues and I did in my Greenberg's lab, Steve Flavell, who's now an assistant professor at MIT. What we showed was that two of the family members, MEF2A and MEF2D, play a role in regulating the density of synapses. So on the right-hand side, under conditions where reducing the levels of MEF2 proteins, we saw an increase in excitatory synapses. And so this would suggest if you're removing it, you're getting more synapses. That's a negative regulator of synapses, or perhaps a promoter of synapse weakening and elimination. So to test that more directly, we generated a version of MEF2. In this case, it turns out to be MEF2C that is fused to the VP16 herpes simplex virus transcription activation domain. So this active form of MEF2 protein, which, like I said, happens to be MEF2C, coincidentally, if we turn that MEF2 on under conditions where we've allowed these neurons in culture, these are hippocampal neurons in culture, to form a very stable density of synapses, and then we turn the MEF2 on, what we found was, as you can see in the yellow bars here, a decrease in the density of excitatory synapses. Not all of them, but a subset. We typically see about 30–40% of synapses, structural and functional synapses, are lost when we activate MEF2 artificially. So we've been working on this, as I said, for about 15 years now. And together we found a number of basic principles that kind of summarized here in this cartoon. And that is that we think MEF is sitting, again, on a number of gene targets within the genome, and it's associated under basal activity states with co-repressor complexes like class 2 HDACs, like HDAC4 and HDAC5. And we know that when neurons are active enough to depolarize the membrane and activate L-type voltage-sensitive calcium channels, this allows an influx of calcium and the activation of calcium-dependent signaling pathways that turn the HDACs off and turn the MEF2 proteins on. And by turning MEF2 proteins on, we then produce RNA and eventually we think protein that ultimately leads to the loss of a subset of synapses. And which synapses and how and so forth and so on has been the focus of a lot of the work over the years. And we've learned a lot of interesting things. For example, that this MEF2 dependent elimination of synapses requires the actions of the Fragile X mental retardation protein. This is the gene, the protein product of the gene that is silenced in Fragile X syndrome. So in individuals with Fragile X syndrome, they lose FMRP function and they have dysregulation of excitatory synapses as well. So we were able to show that these are somehow working in a parallel or linear process. And with work together with my former and current collaborator, Dr. Kim Huber at UT Southwestern, we've been able to show that a number of MEF targets, including the protocadherin gene 10, PCH10, as well as the activity-regulated cytoskeleton protein ARC, are required for MEF-dependent synapse elimination, and those two transcripts are known to bind to FMRP. The RNA is known to bind to FMRP and potentially be regulated its function be regulated by FMRP. In addition, we know there's some other things that are required. We know activity is required both upstream of MEF2, but also required downstream of MEF2 to mediate this process. And some of that we think is through things like glutamate activating receptors like the metabotropic glutamate receptor mGluR5. Again, the details of this are not critical for you to grasp immediately here, but the point here is that it's a complex gene transcriptional program that seems to be focusing in some way on the ability to eliminate synapses. So about 10 years ago now, we shifted our focus away from MEF2A and MEF2D to one of the family members, MEF2C. And MEF2C is a really interesting expression program. It is expressed very highly so what I'm showing you here on the top left is a coronal section of a rodent brain. And you can see that the signal up here, this represents the mRNA expression of MEF2C. And the protein looks very similar, highly expressed in the cortex. Those little whisker-looking things are the dentate gyrus of the hippocampus. And you can see that the expression subcortically is much lower. So we decided to ask, what is the function of this particular MEF2 gene, MEF2C? And again, the reasons for this because some emerging disease connections for this gene. What is the potential function of MEF2C in brain development? So to begin to address this, we generated a conditional knockout of MEF2C predominantly in excitatory neurons of the forebrain. So you can see below using the EMX1 CRE, these are the regions where EMX is expressed, and it's very similar to the regions where MEF2C is expressed. And again, we're only knocking it out in excitatory neurons in this particular mouse. So if you knock out both copies of MEF2 globally, the animals are embryonic lethal. And that's work that Eric Olson showed a number of years ago. The heart does not form properly. The animals are non-viable after embryonic day 9.5. It's involved in the looping of the heart chambers. So obviously we couldn't make a constitutive knockout, and we really wanted to study the function of the brain. So we generated this conditional knockout, and we asked what's going on in these mice. And I'm going to summarize, since we published this several years ago. But the bottom line is these mice, have profound behavioral abnormalities. So they show number of deficits in autism-related behaviors. So for example, they show decreases in social interaction. They show pretty profound decreases in ultrasonic vocalizations, which I'll come back to later, which is a putative communication mode for mice. And they showed abnormalities, if you will, in repetitive and stereotypical motor behaviors. The animals showed really profound motor hyperactivity and really strong deficits in learning and memory, as well as some altered anxiety-like behavior. They showed some reward-based deficits. There were just lots and lots of pretty profound behavioral changes in these mice. So that really clued us in that MEF2C is doing something very important in the mice. And the connection we had with the Fragile X syndrome made us want to pursue this further and understand what may be going on in the brains. And so again, this is a transcription factor. And so one of the first things that we decided to do to try to understand its downstream effects to affect the function and development of these mice was to look at gene expression. So we started by performing an unbiased RNA-seq analysis of cortical tissue from the MEF2C conditional knockouts and their littermate controls. And what you can hopefully see is here on the right, the three group on the right-hand side, we get differentially expressed genes, both up and down regulated. And there were over 1,000 genes that were differentially expressed compared to the wild type controls. The other thing we did was we said, okay, well, what is the nature of these differentially expressed genes? And we compared them to a number of database, transcriptomic databases, including genes that have been linked to synapse development and function, or the so-called synaptome. And at the time, there were about 1,800 of these. And we observed a significant enrichment of MEF2C differentially regulated genes that have been linked to synapse function. And that wasn't entirely surprising, but good to see. The other thing we did is we said, well, what about autism-related genes, considering the social behavior and the communication-like deficits we were seeing. And again, and the connection to Fragile X, again, we saw using, this was at the time, the high confidence Simons Foundation ASD risk genes. And again, we saw a significant enrichment for ASD risk genes that were MEF2C, dysregulated in the MEF2C conditional knockout. And then, of course, a small overlap of synaptic and autism and MEF differentially expressed genes. So this was very interesting and made us think that we were onto something interesting with the connection between MEF and autism and synapse function. And then really to put the icing on the cake, we looked at the Fragile X, the FMRP-bound RNA database, and again saw an enrichment of transcripts that are regulated by MEF directly or indirectly and known to be associated with FMRP. Okay, anybody have any questions before I move on? And I just want to invite you, please don't hesitate to interrupt me. Just speak up. I promise I'll.
And remind me, because I forget, FMRP stabilizes the transcripts, or what does it do again to the transcript?
Yeah, so it's thought to bind to the translating ribosomes. So it binds the RNAs and it slows or blocks their translation. And in response to mGlu-R signaling, you can dysregulate FMRP, you disengage it, and allow for local protein synthesis. So it's thought to be playing a role in targeting mRNAs to synapses and out into the dendrites and poisoning them for more rapid responses to changes in synaptic activity. However, I should say that some of the FMRP transcripts that we've studied that are MEF targets, well, or just FMRP associated, in the absence of FMRP, some of those transcripts, there's more protein made, and some of them it's the opposite. The RNA seems to be destabilized. So it's like probably every biological process. You can't lump everything into one nice tight box. Okay. Oops. Yes. Okay, so we then started asking the question, if these animals have such profound behavioral changes, what might be going on in their brains? And so again, we're knocking out MEF2C selectively in excitatory neurons of the cortex. And so one of the very first things we decided to do was to take an ex vivo slice, which means we take the mouse, we remove its brain, we make a thin slice of the cortex, and then we put an electrode over it and we measure spiking activity. So these are called up states, and these are spontaneously driven synchronous patterns of activity that you see with slow wave sleep. And one of the reasons we chose to look at this is in the Fragile X mice, you see an increase in these up states. And so again, remember the idea here is that if MEF2C is promoting synapse elimination, then if you remove MEF2C, our naive a priori hypothesis was that we would see increased synaptic function in the brain because we're not mediating appropriate activity-dependent synapse elimination. But what we saw was quite the opposite. You can see hopefully here that there was a profound reduction in these up states, which again are this synchronous firing from layer 5 up to the different cortical layers. And this is consistent with a decrease in excitatory synaptic transmission or an increase in inhibitory transmission. So to begin to understand this a little better, we decided to then patch on to the actual cells. And what I'm showing you here in panel B is something called miniature excitatory postsynaptic currents. So we're basically patching onto the neurons and we're measuring their spontaneous changes in current. So these are mediated synaptically. And what we found, just to make a long story short, is a reduction in excitatory, glutamatergic excitatory transmission. So, you know, kind of fitting again with the decrease in the up states. And we saw a reduction in excitatory contacts. So this reduction on the right, this spine densities, dendritic spines are frequently the site of many glutamatergic synaptic connections. And remember, I showed you earlier that when we activate MEF2C, we force it to be active, we can reduce the spine density. So we're getting a pretty robust decrease, which we think is probably accounted for by this mini frequency effect over here.
When did you knock it out? You know, did you try different developmental time points? Because, you know, this is a dynamic process of forming, right, during development, forming synapses, losing synapses. And, you know, if you knock it out or overexpress it, you would expect maybe that both things happen, that, you know, both cause loss because you didn't do it at the right time?
Yeah, well, I think that's part of it. When we look does matter. Here, so we're knocking it out early. So E12.5 is when the Cree begins to express. So we're really removing MEF2C very early on. We're recording here at I think this was P21, as I recall, postnatal day 21. We've also looked at P14, and we've looked a little later. We see something similar, but it is a little bit different at the different time points that you look. And it is a really important point that anytime you're trying to study a dynamic process like this, you can really only take a snapshot, right? You have to kind of start somewhere. And so we chose to pick this period of P21 to P28 because this is the point where there is in vivo, really massive waves of synaptogenesis and refinement going on. But it's a very good point.
I have a question.
Sure.
So the picture shows pyramidal neuron. Is this in cultural neurons, or it's in directly from the brain?
It's a combination. So I'm showing you just bits and pieces of the paper, but the left-hand side, that is an ex vivo slice from the mouse. The lower, the mini frequency, is from an ex vivo acute slice where we patch clamp recorded layer 2-3 neurons.
Okay.
Okay. And then the right-hand one is, I'm showing you cultured neurons looking at the spine density, but we see the same thing with spines in vivo as well.
Okay.
And for reasons that'll become apparent in a minute, we kind of had to jump around between the in vivo and the cell culture model.
Okay.
To try to do some cell autonomous questions. But yeah, good point.
Okay.
And anyway, and I'm not even showing you all the data. We also showed dysregulation of GABAergic synapses. But the bottom line is there is, I should say, a decrease, at least at these time points you've analyzed, a decrease in excitatory synaptic transmission in at least somatosensory cortex where we're looking, and an increase in GABAergic synaptic transmission. And we think the convergence of those two changes are what are giving rise to the really just absolutely clamped up states, the deficit in that sort of network activity, and probably contributing to the learning and memory deficits and all the other behavioral phenotypes we see. So again, the idea that I tried to pitch to you earlier here was that when we activate MEF2, and again, the constitutively active MEF2 was a MEF2c VP16 fusion protein. There's an FMRP-dependent elimination of dendritic spines and synapses. But I just told you that when we knock it out in vivo developmentally, we are getting the same basic effect. We're getting a reduction in excitatory synapses, and in fact, again, an increase in the GABAergic. And so we puzzled on this for quite a while, and it dawned on me that one of the obvious things I wasn't paying attention to is that MEF2 transcription factors, including MEF2c, function as both repressors and activators. And we know that under basal conditions, on many genes that MEF2c is associated with, it is sitting there on the gene associated with the co-repressor complexes that I described earlier. And in this case, presumably, it's sitting there inhibiting genes that would be promoting a change in the EI balance, right? So then if the neuron becomes very active through synaptically driven an eventual memory depolarization, this triggers this calcium signaling pathway that de-represses MEF2c and allows target genes to be turned on. And so MEF2c could either itself switch from repressor to activator, which we know can happen, or the other possibility is it's sitting as a repressor and activity is just removing its repressor function and allowing constitutive transcription factors sitting nearby to turn that gene on. So those are not mutually exclusive. Those are and/or kind of concepts. But the idea here would be that the cell has a potential homeostatic response mechanism that allows for very active cells, presumably during critical periods of synaptic development and refinement, to promote changes in gene expression that would destabilize excitatory synapses and help with synaptic refinement. So there were a couple things we wanted to first establish. First of all, this is an in vivo animal. We've knocked it out in 85% of the excitatory neurons in the forebrain. Perhaps what we're studying is a homeostatic indirect network effect. Well, we still can't rule that possibility out, but we were able to show that if we again cultured MEF2c flox neurons, and again, now these are cortical pyramidal cells in culture, and we transfect a very small percentage, less than 1%, with Cre recombinase to create a knockout such that 99% of the inputs are wild type, we see the exact same phenotype that we saw in vivo and with the EMX Cre mice, that there was a decrease in spine density, a decrease in excitatory transmission, and interestingly, an increase in GABAergic synaptic transmission. So then, oops. So if that's true, if this is a cell autonomous effect, and removing MEF2c is causing this decrease, what about this idea I pitched about it being a repressor? So we decided that if indeed removing MEF2c is dysregulating this repressor function and producing an imbalance by turning on genes inappropriately, if we were to take a knockout cell and put a dominant repressor form of MEF2c, we might predict that this would restore normal EI balance. And I mentioned it and kind of showed you the data already. We already knew that if you take a cortical neuron and you add the MEF-VP16, that you force it to be an activator, that causes elimination of excitatory synapses. So we know that's true. So we knew that we suspected adding MEF-VP16 wasn't going to rescue it, and sure enough, it doesn't. But interestingly, this is exactly what we saw with the engrailed. So we're fusing the MEF2c protein to a strong repressor domain on the engrailed protein, and this reduces MEF-dependent transcription by greater than 90%. In a wild-type cell, you can see here on the bottom right, we have a certain spine density, and if we express the dominant repressor form of MEF2c, we get no change in dendritic spine density. And that makes sense if MEF is acting largely as a repressor, in the cultured system or in these neurons, it's phenocopying what the endogenous MEF2c is doing. Here, though, you can see again in the conditional knockout. So now we're not doing single cell, but we're just culturing all the pyramidal cells from the MEF2c EMX conditional knockout. And again, we see this reduction in dendritic spine density. But if we put the dominant repressor form in, we rescue or normalize the synapse density. So this suggests that MEF is acting in a cell autonomous manner as a repressor predominantly to regulate excitatory and inhibitory synapses. And I'm not showing you the data, but it also rescues the GABAergic synapse density as well. Okay, so again, in the interest of time, because I want to talk about the very recent stuff, is that we've kind of put this into a working model that neurons during a wave of development, synaptogenesis, that they're responding to synaptic activity. MEF2c is sitting on a number of key target genes and repressing their activation. So its important critical role is to keep these genes off until the appropriate time. And then calcium can either switch off the repressor or turn on the MEF2c. This then triggers activation of target genes that in an FMRP dependent manner are destabilizing excitatory synapses and promoting inhibitory GABAergic synapses. And I've just sort of cartoon diagrammed that here on the right, that it would be functioning in kind of this activity dependent feedback mechanism. But in the knockout, the presumed idea then is that genes that should be turned off are now constitutively upregulated, and perhaps the cell is interpreting these elevated gene transcripts as a cell that's very active, and it's promoting the synapse elimination process independent of activity. And we have some evidence to support that idea. Okay, I'll pause again quickly and see if anybody has any questions.
I have one question. So,
Sure.
So how does MEF2c has the opposite effect in GABA neurons?
How does it what?
How does MEF2c has the opposite effect in GABA neurons?
So we don't know what it's doing in GABA neurons. What I mean is that it's in the excitatory neurons, we're getting an increase in GABAergic GABA-A synaptic connections. And that's a postsynaptic excitatory neuron effect. We don't know yet how that's being mediated. We know it requires MEF2c's repressor function, And we had some, I mean, I can guess, but we really don't know yet.
Okay.
But somehow it must be triggering, and it could be a retrograde signal, right?
Okay.
From the GABAergic, from the excitatory cell onto the GABAergic neuron. It could be it also could be indirect, to be honest. I mean, it could be that it's the GABAergic effect is being driven by a change in the excitatory as well. And the other thing, is things like, ARC. ARC can be activated by strong synaptic activity, and it can destabilize neighboring synapses that are not as active. And so perhaps there's some sort of complex code that is feeding back to the interneuron to adjust its contacts. But it's a really good question. I'll kind of touch on that point a little bit later.
Okay.
We know that MEF2c is important in GABAergic neurons too.
Okay.
Like within those cells. If we knock out MEF2c in GABAergic cells, the animals are non-viable.
Okay.
And if we, anyway, I will get into the weeds with that one, but it's playing a role in inhibitory neurons as well. That's kind of a different phenomenon than what we're talking about here, because here we are not manipulating GABAergic neurons at all, right? Okay. It's only excitatory neurons that we're manipulating. Okay.
Okay. Thanks.
So, sorry, did anybody else have another question? I didn't mean to cut anybody off. Okay, let me minimize this. Okay, so, we already had clues around the time some of these papers were starting to trickle out in early 2010 through 2013, that MEF2c was going to be important. It was coming up as a GWAS risk allele for schizophrenia, as for bipolar disorder, and potentially for autism. And but around the time we were working on these projects, a group in Philadelphia showed that there was found a subset of individuals with macro and micro deletions of the 5q14.3 gene that present with a complex syndrome. And when they lined these up, they noticed that all of them had, and most of these have in common, deletion of some or all of the MEF2c gene. And they identified 2 individuals with idiopathic global developmental delay, intellectual disability, and a number of other seizures that had point mutations from exome sequencing in the MEF2c gene. So this led them to coin the term MEF2c Haploinsufficiency syndrome and presume that MEF2c was the gene contributing to this syndrome. Of course, we got excited about this, which is an awful thing to say about finding a developmental disorder, but it suggested to us that we had been on the right track with the thinking that MEF2c was a critical central player in brain development and function. It was originally identified again.
Chris, those mutations loss of function, or were they some other dominant negative effect like that mouse you made?
Yeah, well, that's a good question. I'll come back to that.
I didn't mean
No. It's actually interesting. So they didn't know. They just had the 2 point mutations, and you'll see they're super conservative. They found the 2 missense mutations and they had no idea. Which led me to, you'll see in the next slides. So, again, this was originally was identified as the probable critical gene within the 5q14.3 microdeletion syndrome. And one of the interesting things, MEF2c is expressed not only in the brain, but is also expressed in the immune system and is expressed in the muscle and cardiac muscle as well as skeletal muscle. It could presumably contribute to development in a number of ways. But interesting, most of the symptoms seem to be nervous system, neurological in nature. So for example, most of the common clinical findings so far are that patients have severe intellectual disability, most have autism spectrum disorder, most have seizures by the age of 2, most of them have an absence of speech, some will have a few words. They also have motor problems like limited walking abilities, hypotonia, and a lot of stereotypic movements that you see sometimes with kids with autism, but also that you sometimes see with pretty severely affected neurodevelopmental and severely neurodevelopmental disorders. There were some minor brain malformations reported from some fMRI studies, but they were inconsistent, and so there's no consistent pattern of structural changes in brain. So as Larry alluded to, this really made me start thinking about, well, you know, sure, we're knocking out the MEF2C gene in these microdeletions. Are they sure that MEF2C is contributing to this? And what is the contribution of MEF2C to this phenomenon? And really the best way to get at that is to think about the point mutations, because the deletions are taking out, and sometimes 20, 30 other genes or more in some cases. But the point mutations give you a real opportunity to really think about the actual role of MEF2C in this process. And so as you can see in the upper panel, again, here's the coding sequence for MEF2C. What we're able to do is we teamed up with a group at the Greenwood Genetics Center here in South Carolina, and I was giving them a talk similar to this about 5 years ago. And I said, they sequence a lot of kids in South Carolina that are referred by their pediatrician because of global developmental delay, seizures, and other types of symptoms like I described earlier. And so they looked in their database and they found 5 kids in South Carolina who had point mutations in MEF2C, and those are shown here in green. As well, we teamed up with, I found a group online, a family group of this MEF2C haploinsufficiency syndrome, and they were very generous to share their kids' missense mutations, or as you can see, a lot of premature stops. One thing you'll note is that almost all of the missense mutations cluster in the DNA binding and dimerization domain, whereas most of the premature stops with the X or the frame shifts, that all of these frame shifts that are marked denoted here all cause a premature truncation. There are no point mutations outside of the DNA binding domain, except for this one that we just found about a month ago. And this is a mutation that we find in the nuclear localization signal, which I think probably makes a lot of sense with dysfunction. So we then decided to make these mutations that do exactly what Larry was referring to. Are these really loss of function mutations, and or could they be acting as dominant negative or dominant autosomal dominant effect, or is it truly haploinsufficiency? And the point I wanted to make about the reason why it's really it was really cool to be able to do this genetics on the human population, because we could just look at the sequence conservation from yeast to humans, and you can see that the DNA binding domain is highly, highly conserved, as is the DNA, the dimerization domain, whereas you lose conservation outside of this region across species. So we would have guessed, knowing nothing else, that mutations in this region might be very important to its function. Now, again, if you look at some of these mutations are really, really modest, like the G27A, a glycine alanine mutation, you might consider to be a super conservative mutation that would not have an effect. So to try to get at this question, we made a handful of these mutations, and we tested them for their ability for the patient mutants to bind to DNA. And we used an assay called an electrophoretic mobility shift assay, or EMSA assay. And basically what we do is we take a piece of DNA that contains a MEF binding site, and we label it so that we can detect it. And we run it together, we incubate it together with a lysate expressing MEF2C, either wild type, which is shown here, either wild type or the various patient mutants. And if MEF2C dimers bind to the MEF2 piece of DNA, it retards the migration of the free DNA, and that's what you're seeing here. And you can see that even the G27A, which is, again, a very, very conservative mutation, disrupts the ability of MEF2C to bind to DNA in this assay. And every single one of the patient mutations that we have tested so far, including the ones I'm showing you here, all of them either disrupt DNA binding and or destabilize the protein so that the protein is knocked down 90%, probably due to structural problems with folding. So this suggested to us that these mutations that we're seeing in the patients are indeed loss of function. In time, to answer Larry's question, we then ran these against wild-type MEF2C to ask whether they might be acting as a dominant interfering form. We see no evidence of their ability to compete the wild type MEF2C to bind. So we think it's just strict haploinsufficiency and not a autosomal dominant, at least using these types of assays. Okay, so if MEF2C loss of function or haploinsufficiency is associated with disorder, can we model this in a mouse? And so we took advantage of a floxed allele of MEF2C that we had, where LOXP sites were flanking exon 2. And exon 2 codes for a piece of the DNA binding domain and the dimerization domain. And fortunately or unfortunately, when you remove this exon 2 in CRE-mediated recombination, it splices in frame from exon 1 to exon 3. So you make the entire protein minus this little piece of the DNA binding domain. So we decided, well, this was a useful model. Remember, in humans, it's haploinsufficiency. You have one wild type copy and one mutant copy or deleted copy. So we first made this mouse, and it's viable, although there is some lethality. We don't know exactly where they're dying, but it seems to be embryonic. About a third of the MEF2C global heterozygous mice are lethal. Here's the wild type protein running up here, and you can see that this is the truncated version of the protein that would correspond to this, that you can see drops down in size, and there's some fragments generated probably from degradation or instability. And then, of course, we validated that indeed this mutant form of exon 2 does not bind to DNA, and in data I'm not showing you, if we overexpress it, it does not compete away the wild type MEF2C's ability to bind. So we don't think it is, we don't think this delta exon 2 mutant protein is able to function as a dominant negative. Okay, so the mice are mostly viable, as I mentioned, and they look relatively normal physically. So we wanted to ask what might be going on with some of the behaviors. And so we decided to focus on some of those autism core symptoms, social deficit speech, like deficits using communication, repetitive behaviors, as well as some of the other symptom phenotypes that we would see in humans. And so I'm going to show you, just highlight a few of the things. Again, we just published this a couple months ago, so this data, you can look into some of the details. One of the things we looked at was the social interaction assay. And the way we do this in mice is we have 3 chambers, and the test mouse is placed in the middle. And on one side, we have a cage that has a conspecific, novel, same-sex animal. And on the other side, on the same type of cage, we have a inanimate object of the same size and shape. And then we let the wild type mouse, or the wild type or mutant mice, interact for a period of time, and we measure the time they interact with the social target versus the non-social target. And what we saw is that in the control animals, the littermate, wild type littermates, we see that they show the very typical preference for the social versus the non-social object. And so this is what we typically see and what you would expect to see from a control. In the MEF2C global heterozygous mice, the MCHS model, we see that they lose this social preference. They're still interacting, but they're not showing the typical preference for the social animal. We also looked at a species-appropriate putative mode of communication, looking at something called ultrasonic vocalizations. And some of you have probably heard of this. Mice will generate this high-frequency calls that's above what the human ear can hear, and they generate these calls. I'm showing you here a sonogram of these calls. They'll generate these squeak-like calls at frequencies between, in these social contexts, between 50 and 100 kilohertz. Humans can hear up to about 20, 22. So we use this special microphone, and we measure the vocalizations that are generated by the pups. And again, similar to what we might have predicted, and similar to what we saw with the conditional knockout in the earlier study, we see a very profound reduction in the generation of these ultrasonic vocalizations. Now, in this assay, we're separating the pup from the mom. So the pups are calling to the mom, presumably be retrieved, and we're getting a dramatic suppression. We also see this in the adults in a different type of ultrasonic vocalization assay, where we're taking sexually mature males and putting them in the presence of a sexually receptive female. And what happens when you do this is a male will go from making 0 to 1 call in 5 minutes to making hundreds of calls, often, even as many as 1,500 calls in the presence of the female. And again, we saw this dramatic reduction in the generation of these ultrasonic vocalizations. So we then moved on to look at some motor behaviors. And interestingly, we saw a sex-specific effect here where the males show a profound motor hyperactivity. So we place them in these activity chambers. They're roaming around, and we can monitor their activity both horizontally and vertically. And again, similar to what we saw, the brain conditional knockout, if you will. And we also saw evidence of a repetitive type behavior where they go to the corner and do this repetitive jumping, which is something we also saw with the EMX conditional knockout. And as you probably know, repetitive restricted behaviors are fairly common in autism, at syndromic and non-syndromic autism. One of the things that we looked at that was a little disappointing, if you will, from trying to generate an animal model of MCHS was that in humans, they have a profound intellectual disability. And so we tried to look at learning and memory tasks in these animals. And we really so far have seen almost nothing. So we've looked at classic fear conditioning. We've done spatial learning and memory in the Barnes maze. We've done novel object recognition. We've done Y maze for spatial working memory. And then we've done a really more cognitively demanding task, which is operant self-administration of sucrose. So the animals have to press an active port to get a sucrose reward. And it's a really rich and robust assay where we can look at their ability to discriminate and learn the active versus inactive port. We can look at the time it takes them to learn the assay. We can look at extinction learning when the reward is no longer present. And we can even look at long-term memory associated with the cue that was initially linked with the active lever and the reward. And none of those things seem to be affected. So that was a little disappointing, and perhaps it's intellectual disability and mouse learning and memory are not so connected as we'd like to think, or that this represents a rodent-specific function for MEF2c, where humans are a little more dependent on MEF2c function. One of the things that's interesting in humans with human patients with MEF2c haploinsufficiency is they show very high pain tolerance, and actually see this quite commonly in a number of neurodevelopmental disorders. And a lot of the parent groups had been telling me about difficulties with their children, not sensing pain, sitting in an ant mound and getting bitten and not seeming to notice or react, laughing when they get their shots, inoculations, when they're children, et cetera. So we decided to look at this by using this special chamber where we can place the animal in this little box, and it's sitting on top of a force plate. And when the animal receives an electrical shock, the animal flinches, and we can use a very nice sort of dose-responsive analysis of the shock-response relationship. And you can see the control animals that the bigger the shock, the bigger the startle response. And in the MEF2c heterozygous mice, they do respond a little bit, but it's pretty flat and a pretty profound deficit in this, what we're calling sort of acute nociceptive sensitivity. We still haven't quite figured out exactly what the cause this is, but could represent central processing of pain, or it could be representing some sort of peripheral nerve function. So we looked at a bunch of different things. Some of those I'm showing you here. A lot of the things that we looked at and would have expected to see from common MCHS symptoms, we do see something similar in a mouse. Some of the things, again, the learning and memory seem to be not abnormal. We do see changes in sleep patterns, which is another problem that the kids see. We just published that recently in eLife. Okay, so if these mice represent, at least at some level, some dysfunction as behavioral dysfunction that might relate to MCHS, what about what's going on in the brain synaptically? And so just really briefly, there's a whole several figures on this in the paper if you want to follow up a little more in the detail. But to make a long story short, we see something kind of similar to what we see with the conditional knockout, which is that if we record from layer 2-3, and here we're looking in somatosensory cortex, and we stimulate collateral fibers, these are horizontal fibers coming in from a neighboring barrel column, we get a reduction in the amplitude of the excitatory response. So we're seeing a reduction in excitatory responses. We're seeing a reduction in upstates that I'm not showing you. And interestingly, in this case, we're actually seeing a increase in paired pulse facilitation. So this is interpreted as a decrease in presynaptic probability of release, which is probably contributing at least partially to this deficit. However, we also see, if we just patch on again and record, we see a decrease in the amplitude of the miniature responses. So again, without getting into the weeds, this is a consistent again with a weaker excitatory synaptic response in the global MEF2c heterozygous mice. And in this case, we're looking at postnatal day 35, about 5 weeks old. I'm not going to get into this, but this is a really interesting thing as well that I'll, if we can come back to talk about later. Okay, so since this is the genomic genetics group, I thought I would again talk a little bit about the genes, because ultimately we think that's what MEF2c is doing to produce these abnormalities. In this case, again, we're looking in cortex, and in the heterozygous mice, we're seeing this time about 500 differentially regulated genes. And again, about half of them are up and about half of them are down. We don't see a difference by sex. And here with a volcano plot, you can see a subset of the genes, some of the biggest changes. Some of these are quite large and some of them are more modest, but nonetheless, a pretty robust group of dysregulated genes. We did some kind of similar analysis that we did last time, where we were looking for enrichment of genes that are dysregulated in the MEF2c heterozygous mice. And again, we saw enrichment for FMRP-associated RNAs. And again, we saw enrichment for autism-regulated genes, genes that are dysregulated in autism, particularly those that are downregulated in autism. And so that was kind of interesting as well, that we're seeing this, that the downregulated genes MEF2c are related to the downregulated in autism. The other thing we decided to do is look at sort of the rich databases that have been coming up from human transcriptomic analysis. And this was from the psych and code modules. And what we did is we then just plunked in again the differentially expressed genes, the MEF2, and we asked where is there enrichment with the psych and code module. So again, these are modules of genes that are dysregulated in human disorders. And there's a bunch of them here, and I don't want to go through it, but it's autism, schizophrenia, and bipolar disorder, and a number of things. One of the things that popped up with enrichment of the MEF2c enriched genes, and particularly downregulated, are genes that are downregulated or dysregulated in excitatory neurons in individuals with autism. The other thing we saw that was fascinating was a dysregulation of, or an enrichment for genes that are dysregulated in microglia in individuals with autism. And those tended to be the upregulated genes that we were seeing for MEF2c. So they're upregulated in autism, idiopathic autism, and they're upregulated microglia-related genes in our MEF2c heterozygous mice. So we thought that was really fascinating and really started thinking about the fact that for this disorder, can we pinpoint which cells are most important? And so if we looked at MEF2c expression in the brain, so now I'm showing you a sagittal mouse section, you can see again, screaming signal of MEF2c in the cortex, in the dentate gyrus, a little bit in CA1, this is actually interneurons and microglia, a little peppering of interneurons throughout, it is expressed. And these are really regions, and I don't know if you can see this well on yours, I can see it on mine, but you can see the Purkinje cells of the cerebellum, also very highly enriched for MEF2c expression. And so just listed here, you know, MEF2c is highly expressed in cortical pyramidal, hippocampal dentate granule, GABAergic interneurons, Purkinje cells, and you can also see the olfactory bulb. So we could take advantage of the fact that we have the Flox mouse, and we can do some genetic dissection by removing one allele and asking if we can recapitulate some of the behavioral and synaptic phenotypes. And indeed, again, we just looked at the 6 behavior changes that we saw in the global heterozygous mice. And if we remove one allele of MEF2c from the EMX lineage, so again, this is forebrain excitatory neurons predominantly, we don't see the social and communication-like deficits, But we do get the repetitive jumping in males. We do get the male-specific hyperactivity, and we do get this altered anxiety-like behavior that I didn't really show you. But we don't get all of it. So neurons are important. We looked at parvalbumin Cree mice because PV is a population that MEF2C is highly expressed in PV interneurons as well as other interneuron classes as well. And if we knock out one copy of one allele of MEF2C in the PV Cree line, the only thing we see so far is a pretty modest effect on hyperactivity. The rest of the things, rest of the behaviors we've analyzed seem normal or typical. We also knocked out a copy of MEF2C in the Purkinje cells of the cerebellum, and I was really thinking that cerebellum's linked to autism and it's linked to vocalizations, and we didn't see it here, so we were going to see it here. But unfortunately, we really see absolutely nothing no behavioral abnormalities in the Purkinje conditional heterozygous mouse. Okay, so what's contributing? So we're presuming that most of this is being driven by the neurons, but there are other cell populations contributing to this. If we looked at RNA expression in early development, we noted from a work from Ben Barris's group that the expression in neurons is almost as high, if not, is about as high as it is in microglia. Interesting also endothelial cells. And so this really led one of my students, Catherine Bridges, an MSTP student in the lab, to ask whether MEF2C might be playing a role in microglia. And it's expressed in microglia, and we showed that throughout the brain, and it's expressed during development. So what are microglia? As you probably all know, there's the brain's resident immune cells. They play important roles in embryonic development. And within the brain, they play roles in regulating survival and death. But the thing we were really excited about was, the well-known work from several groups, including Beth Stevens and Barris and others, that microglia play an important role in synaptic remodeling and plasticity and pruning. And again, we were thinking that perhaps MEF2C, through its role in excitatory neurons, might be mediating this, but perhaps it could also be playing a role in the microglia. And so one of the things that Catherine did was she analyzed the brains of the global heterozygous mice, and what she saw was a significant increase in IBA1 expression without a change in soma size or the density of neurons. So we're just increasing the expression of IBA1 in the MEF2C global hets. So that led us to think, well, perhaps this increase in IBA1, which is a marker of microglial activation, maybe we've got hyperactivated microglia, and that they are doing things they shouldn't be doing. And we still don't have the answer to this, although I can tell you that if we just take mice out of their home cage and we analyze neuroimmune markers, we don't see strong or clear evidence of like basal hyperactivation. So we think some of this increase in IBA1 is transcriptional, and that some of those other transcription regulated genes. So we do see upregulation of interesting genes like C1q and C4, and a number of complement factors, which we think may be mediating this. So if MEF2C is dysregulated, we're removing a copy of MEF2C in the global het, if it's playing a role in microglia, then we predicted that if we were to remove one allele of MEF2C selectively in microglia, that might produce behavioral effects that are reminiscent of what we'd see in MEF2C haploinsufficiency syndrome. So what she did was she generated a microglia selective conditional heterozygous mouse, and I won't go through the details here, but we made a very early postnatal heterozygous in the microglia, and then we looked at behavior and synaptic changes. So what we saw in behavior was interesting. So things like the anxiety-like behavior didn't seem to be affected. The hyperactivity didn't seem to be affected in the males, but we were able to recapitulate and this increase in repetitive jumping. It's not as strong as it is in the EMX, but it's there. And then the other thing that was really interesting was that we saw that if we remove a copy of MEF2C from the microglia, that we are dysregulating social interaction, social preference. So it's interesting that there seemed to be an enrichment for autism-related behaviors when we're removing MEF2C selectively, one copy of MEF2C selectively from the microglia. The other thing that was super fascinating as well was that in addition to these behavioral changes, removing one copy of MEF2C from microglia was also sufficient to create changes in excitatory synaptic transmission. So again, remember, we saw this with the global heterozygous mice. Well, we see it again simply by removing a copy of MEF2C from microglia. And so we think this may be an indirect or direct consequence of microglia function to regulate synaptic connectivity. And we're following up on these studies now to figure out exactly how MEF2C might be mediating this effect using single-cell RNA-seq and a number of other approaches. Okay, sorry, I went a little long. The basic summary is that MEF2C is playing a role in controlling synapse development, and we think that as largely as a cell-autonomous transcriptional repressor, and the deletions and mutations of MEF2C is associated with MCHS, MEF2C Haploinsufficiency syndrome, and that we can mimic some aspects of that by creating a global MEF2C heterozygous mice. We see changes in synaptic transmission as a result of loss of one MEF2C allele, and we see that changes in gene expression produced by MEF2C haploinsufficiency are linked to excitatory neurons in microglia, and that we can attribute some many of the behavioral and synaptic phenotypes in this mouse model of MCHS through roles in both neuroimmune and neurons. And I have to thank thank the team that contributed to this work. Obviously, I get the pleasure of talking about the findings, but all this hard work was done by a group of talented folks in my lab, including Adam Harrington, Catherine Bridges, my electrophysiologist, Evgeny Svetkov, and then Jennifer Cho, an MSTB student, Alema Sali, my lab technician, and some helpers from the College of Charleston. We've got some really important collaborators, Steve Skinner and Hannah Warren at Greenwood Genetics Center, some colleagues here at MUSC that have helped us as well as some bioinformatics assistant from Jenna Kanopka and Stefano Berto at UT Southwestern. And with that, I'll take any questions.
I'm going to raise my hand. So Chris, thorough, beautiful work, detailed, extensive. I mean, you know, you did a good job, especially the behaviors. I think, though, you may want to consider that although you were modeling one disorder, pretty successfully modeled autism. You know, yeah. I mean, what you have is an autism mouse, really. Let's be honest. It has all the features of an autism mouse, even the male preference for certain autism behaviors, you know, the FMRP involvement. Like, you know, you might want to consider that people and mice aren't the same and that you've got a decent autism model to study, right? That's just one comment. And you might want to say something about that. Maybe, I'm sure you've thought about it since you're funded by Simons. But.
Yep. Yeah, no, absolutely. I mean, the whole reason we got into this was originally the connection between MEF and FMRP and syndromic autism. I think that while I agree, and I think the reason we're seeing such robust autism-like phenotype, I mean, we're seeing it's autism plus, right? Which is what MCHS is, right? It's autism plus. It is.
This is an intellectual disability disorder. You can't mix intellectual autism. It gets confusing for people, but the truth is, if you want to work on syndromic autism, like I've done with Dept15q, then these kids have normal IQ. You know, like that's one of the features, and your mouse seems to have normal IQ. So. Yeah,
although, you know, I quibble a little bit. So, you know, I mean, you know this, like 40% of kids with autism have intellectual disability. And while we like to think of the high functioning end of it, almost half of individuals with autism have intellectual disability. So, yes. You know, it's hard to separate.
But that's
autism is not a clean,
autism, right? You know, now we're talking about, I don't know what caused this autism, as opposed to syndromic autism, which you're on a track. I've been on this track and still sort of on this track. You know, the idea being that if we can figure out syndromic autism, we get to the molecular changes.
Yeah, right.
In general, right?
Yep.
As opposed to, I don't know what autism, which includes intellectual disability. I mean, that's a debate for another time. But I just wanted to point out that what you know, I'm sure that these features are very specific. I mean, you have all the right features in your model to have an autism model.
Yep.
And the other unfortunate thing is that I think I like your microglia, neural immunity stuff. The paper just came out, somebody sent to me, about the immune system regulating social behavior in mice. I don't know if you saw that.
Yep.
But, that's kind of a game changer that people didn't really consider.
Right.
That maybe tweaking the immune system could cause 3-chamber test effects that they hadn't even considered in mice.
Yep.
And so I guess the actual question then is MEF2 involved somehow in the immune system as well, and immune system T-cell development or something like that?
Yeah, so MEF2c, I mean, microglia obviously is part of the immune system, right? I think that MEF2c has also been shown to play a role in peripheral T-cell development. I think the way that we made our conditional knockout, we're not affecting T-cells because we do it early and those turn over, whereas the microglia stay resident. But you're exactly right. And I think that actually some of the patients with MEF2c haploinsufficiency, they have trouble regulating fever. They have some of them have febrile seizures. And I do think many of them struggle with immune related. I completely agree. I think some of the phenomena is going to be linked to immune system dysfunction. It's something that we're looking at a little bit in collaboration with some friends of mine here in the micro and immuno department. Really sort of staying more focused so far on the brain immune side of it, but yeah, absolutely. I think you're exactly right. It's certainly possible that some of this is coming from indirect effects of immune system dysfunction. And actually related to that, I think one of the things that's kind of interesting about the microglia piece, right? So when we look at the differentially expressed genes, we are seeing this upregulation of microglial genes as a population. They tend to be more upregulated than downregulated. And we're seeing C1q, B, C1qc, C4, these complement system factors, as well as some other factors that are involved. Microglia are also involved in pruning oligo precursor cells and doing lots of things in early brain development and in ongoing brain maturation. And MEF2c just came out as a GWAS hit for MEF2c SNPs came up as a GWAS hit for Alzheimer's disease. And one of the ideas that's being kicked around, and I think there's some probably truth to this, is that perhaps if you have hypofunctional MEF2c, if you have a SNP that decreases your MEF2c function, that you have an overreactive neuroimmune system, and that this is leading to premature pruning and over pruning and synaptic remodeling during age that may contribute to its association with AD. But these things are so intertwined. I mean, I agree. It's hard to make and again, I will be the first to say that a mouse is not a human, and that we all try very hard to make our mice, pound that square peg into the round hole. And I would even go farther and say that even a deficit in social interaction in a 3-chamber assay is, kind of a crude measure and could be accounted for by a number of different things. There's a million controls you have to do to show that really is social.
I'm with you, man. I mean, I've written enough grants about Drosophila that I can tell you flies aren't people either. And,
they're not?
No, not really. But it makes it, it does make it difficult. But when you find, serotonin and dopamine signaling are affecting your phenotype, it's not a big leap, in my case, to say that just like human neurons, they have the same basic processes, which is why it's complicated to do behavior in any animal model. We're doing behavior, but it's seizure. It's crazy, on-off kind of behavior. When you start doing social interaction, which we actually have also done in flies, it becomes a little more complicated to make that leap to humans do this and flies and mice do this. I mean, I would never say that that's a fact either, but it leads you somewhere sometimes.
Yeah, absolutely.
But very nice. I'm glad I came. I zoomed.
Yeah, the altered behavioral effects that you saw that were largely driven by males, is that specific clinically as well? Do you also see that clinically, or is that just randomly what you saw in mice?
Yeah, that's a really interesting point. So for many of the symptoms of and the prevalence of MEF2C haploinsufficiency syndrome, and they're showing up in the clinic with developmental delay, there's not a bias between males and females. For the autism component of it, it is more common in the males than the females, and so maybe that's why we're seeing basically it's the repetitive jumping and the social behavior that tends to be a little more, it's stronger in the males than the females. Actually, the hyperactivity is, for that matter, as well. It's not a clean connection with the humans, though, for sure.
Okay.
So I was a little bit puzzled. So I think, and maybe you can clarify this. So it seemed like initially you were talking about an EMX-driven knockout in the first part of your talk that did show cognitive deficits.
Yeah, so that's when we're knocking out both copies. So there's
oh, that's both copies.
Yeah.
Okay.
So and that's something to talk about. Larry's point as well. It's interesting. Sometimes in a lot of the mouse models of autism that are out there, they actually will knock out both copies of the gene, and that it's only then that they see social deficits, repetitive behaviors, whatever, and that the Het, which genocopies the human better, right, has better face validity or construct validity, doesn't show anything. And so I've often pondered the idea that maybe humans are just more sensitive to gene dosage changes, and mice are less sensitive, and they need, you need to remove both copies before you get a super robust. Now maybe MEF2C escapes that to some extent, because it is impacting, it's a transcription factor, it's a hub gene, it's affecting hundreds of thousand genes. Maybe we're able to see an effect there so easily because it's such a central player. But again, wild speculation. But like Larry, I've been doing this stuff with the autism space for a long time, and often when you see people give talks about autism genes, they knock out the gene. They're not generating a heterozygous mutant, and that's when they see an effect finally. But in humans, you almost never see that. The only exception is Fragile X syndrome, because it's X-linked. Males are affected, and so obviously you get a Y X, and so you actually get a nice clean knockout that connects to human patients.
Well, the copy number variant disorders are also heterozygous, if you think about it.
That's right. Yep.
So, and those are major drivers of autism in any clinical cohort.
That's right.
Ask your friend Skinner, who I've actually met before.
Yeah.
Randomly look through the cohort, and you'll find 5% of them are copy number variants, 15q, 16p.
Yep.
Dosage.
But but why is it that mice seem to be able to tolerate the gene dosage more easily? And is it because the brain is a little simpler? Is it because they have compensatory mechanism, or is it because through human evolution, the complexity of the neural circuits and neural function is so much more complicated and sensitive to subtle changes? And the mouse, you really got to give it more of a sledgehammer.
Yeah.
But it's also possible that the mouse may be abnormal. We just cannot tell.
Yeah, that's right. That's exactly right. That is also very, very possible. That, particularly like intellectual disability, right? I like to talk to the parents about this, because human intellectual disability is not an animal not learning to freeze when it's getting shocked in a chamber, right? I mean, that's a pretty discrete and robust stimulus that you can be really intellectually impaired as a human and still figure out when you're, in fact, a lot of these kids, they're severely intellectually disabled by sort of clinical definition. But as soon as the parents tell me this all the time, as soon as they start wheeling the kid toward the doctor office where they get poked and prodded, they start screaming and crying. They have made that contextual association very, very robustly. So the fact that we don't see deficits in our learning and memory tests in the mice, to me, just says that they're not sensitive enough to pick up what maybe really is the underlying problem.
In the words of Jim Lupsky, even though he wouldn't say it now, probably.
What's that?
In the words of Jim Lupsky, who probably wouldn't say it now, but he used to say this, how can you tell the mouse is retarded? I say that all the time. Because he's right. He's like, you know, you can't interview him. I mean, you're making up these behavior assays that may or may not mean anything at all.
That's right.
You know?
Well, the other interesting sort of clinical point to raise, I think, is that the parents tell me that the kids are so profoundly affected, right? They have motor problems. They have, you know, they're distracted by a lot of things that are going on. That may not be an intellectual ability problem, but an attentional problem, right? They're distracted, or they don't have the physical ability to, and a lot of them will have, you know, they'll have eye problems.
It'd be interesting to compare their, you know, when you first described this syndrome, which I'm not familiar with, you know, it sounds actually quite a bit like Angelman syndrome, which is funny.
It is.
They really have a lot of the same intellectual and communication defects, and what sound to me like more balance than motor defects, right? I mean, that's, you know, like Angelman kids have trouble going through doorways, right? Like when they can walk. And it has more to do with information processing than it does with physically not being able to move.
Yeah,
right?
yeah.
And it sounds like they had some of these, I don't know, I haven't seen any of these kids, so I don't know.
Yeah, a lot of them are in wheelchairs and have major, actually interesting that the kids with point mutations are more likely to have independent walking, are less likely to have seizures, you know. So I do think that the deletion, the copy number variant, that is a multigenic.
You don't even have breakpoints. I mean, I don't know. I have to look up the syndrome, but your breakpoints are inconsistent, right? Like you've got big chunks, medium chunks, little chunks. Yeah, there's overlap, but to call it a syndrome and there's like 30 genes, 15 genes, 10 genes, 8 genes, those are all different syndromes, you know? Like you can't really assume that those are the same. I mean, I would agree with that.
Yeah, and also the N is really low. I think right now what they're doing is there is a 5q14.3 syndrome, Haploinsufficiency syndrome, and then there's MEF2C Haploinsufficiency syndrome. And I think part of the work that we were hoping to do is to really define, like, what is the MEF2C contribution?
What's
and
the
is it
MEF2C contribution?
You know what I mean? Like, what is the MEF2C contribution? Because I think that the point mutations, the point mutants, and the premature truncations, that's really going to define what is a true MEF2C phenotype. Another little interesting tidbit for those of you who are not bored to death right now with the weeds discussion here, is that you remember that G27A mutant I showed you? So that was in an individual in New Zealand. And I saw that publicly, it came out in that very first MEF2C haploinsufficiency together with the 5q deletion, where they first started calling it, you know, as the candidate causal gene. I saw a list of, you know, we got a list of some individuals that have been kind of coming in, and one of the parents emailed me their genotype, and it was a G27A, and I just assumed, right, it's got to be the same kid. And several months into the study, I finally made a connection. Like, this one was coming from somebody in Ohio, and there's an individual in New Zealand, completely unrelated, who had the exact same amino acid. I mean, I think the nucleotide is a little different actually, but exact same mutation. The chances of that are incredible.
Yeah.
And both are severely affected. So, I mean, you know, it's real.
I remember you showed a picture that shows that the superficial layers have more MEF2C. So do you see at a cellular level this layer-specific effect?
Yeah, that's interesting. So what it can tell you is that it depends on exactly when you're looking at development, but after the cortical plate, you know, MEF2C is expressed very early as layers 2, 3 are migrating and developing. So right after they differentiate, layer 4 expresses MEF2C very highly. Layer 5, if you look in, you know, it depends on when you look. Layer 5 has a really low MEF2C expression, and then layer 6 has sort of an intermediate expression. And the reason I think it's kind of fascinating is one of the targets that we found. So mostly we've been recording from layer 2, 3, right?
Okay.
But we've also recorded from layer 5. And we record from layer 5, we also see the decrease in mini EPSC amplitude. No effect in frequency. So it's, you know, obviously because you see a differential expression doesn't mean that it's not there, right? It is expressed in layer 5. It's just lower than it is in layers 2, 3, 4. But what was fascinating is when we looked at the MEF2C conditional knockout and we looked for genes that were going up. So the simple idea is MEF is repressing some of the genes. What are some of the genes that are upregulated when you remove it? And one of them is a protocadherin, not protocadherin 10, but a different one. And if you look at the expression pattern, and it was upregulated like 3-fold when you remove MEF2C. If you look at an in situ hybridization of MEF2C, highly enriched in 2, 3, and 4, almost absent in 5, and intermediate in 6, the PCDH gene has the complete opposite pattern. And so that's one of the targets that we've been following up on that we think may be coding sort of a cell adhesion component that may be affecting, because we can phenocopy some of the findings simply just by manipulating this protocadherin.
Makes sense. Great.
But let me add one more quick thought, which the other thing that's interesting is one of my colleagues, Kim Huber, did this experiment where they cell autonomously knocked out MEF2C on one side of the cortex, okay? And they did it in a fairly sparse manner. If they patch those knocked out cells and they stimulate neighboring cells, so either layer 4 or layer 2, 3, they saw a reduction in evoked responses. Okay? If they do minis, they see the same thing we do with the global Het. There's a decrease in amplitude and an increase in frequency. Well, the increase in frequency is super complicated, right? That's presynaptic perhaps, although if anything, we saw a decrease in presynaptic release. So the other idea she had was maybe these were long-range connections.
Yeah.
And indeed, if she stimulates contralateral cortex and looks at evoked responses, she sees an increase. So we think one of the things that MEF2C is doing is it is coding for layer-specific hemispherical connections.
Okay.
And I think it's really kind of interesting and fun to think about the idea that MEF2C is coming on and affecting local inputs and long-range inputs, and that in the visual cortex, for example, you kind of sometimes see the same thing where you see an increase in frequency and a decrease in amplitude if you do an activity manipulation.
Okay.
And the reasoning, the thought behind that is that during synaptic refinement, you're getting weak synapses and strong synapses forming, and then failure to eliminate is causing a net reduction in mini amplitude, but an increase in frequency. And so, you know, kind of trying to tie back to that synapse elimination idea, one of the things we're thinking about now is that maybe what's happening is that we're getting, we're maintaining weak local synapses that should have been removed.
Okay.
And that is messing up the dynamics of cortical activity.
Makes sense. Great. Good work.
Thanks.
Good work.
Graduate students, so we're in kind of like a 45-minute question and answer period. And are there any graduate students that want to ask questions, or you can just introduce yourselves? Up to you guys. And that's true of any of the people who stuck around. Feel free to introduce yourselves if you have questions. Crickets. So I guess one of the things that I was really interested in the microglial part of your story. And I think you were showing data from maybe like day 30 or 40 when you looked for changes in microglial morphology and expression. And it wasn't really clear what the microglia were doing, but you mentioned that maybe they might be primed. Did you look at as the mice age, were there any changes in that?
Yeah, no, that's an astute point. So we've looked at P35 and we've looked in young adult, and we see this consistent upregulation in IBA1. We don't see the classic, like just in a home kit, we don't see the classic activation profile, at least morphologically. However, if you look at TNF alpha, if you look at TGF beta, if you look at IL-6, and they're up. All of them are kind of consistently up, but not to the level that we can, not statistically significantly so. So it makes me think that there's a little bit of sort of trickle hyperactivation of the microglia. The other reason that I think that that's going to turn out to be true is that when we give animals an injection of LPS, when we give animals an injection of LPS, what we see is the wild types induce microglia, and we see that we again exaggerated activation in the MEF2c-Hets. Again, some of this stuff is still work in progress, but the reason I mention it, and we haven't looked at a bunch of different ages, we've just looked at one age so far. The reason I mention it is that I got really excited about this microglial finding early on, and I thought, well, if we have this sort of brain on fire thing going on, where you've got this hyperactive neuroimmune system, then if we could dampen that down, perhaps it could ameliorate some of the phenotypes in the mice. And so we're almost finished with a minocycline treatment regimen. So these animals receive minocycline from P2 all the way through adulthood. They get them from mom first, and then through their drinking water. And it does look like it is able to rescue, again, interestingly, in a male-specific fashion, a subset of those phenotypes. I don't know that it's related necessarily, but it's intriguing. And other mouse models of autism have also shown, some effects of minocycline. I don't think minocycline is going to be the magic bullet to cure MEF2c haploinsufficiency or autism, but I do think that we're on to something about overreactive neuroimmune system. The other thing,
whatever it is, though, you're going to have to do it early, right? Like Mark Zilka gave a really good talk here recently.
Yep.
And he was talking about in utero treatments for Angelman syndrome, right? Because in those mouse models, you can actually tell that if you don't turn UBE3 on early enough, that you turn it on later and nothing happens. You can't rescue it. So he was proposing that, we do these in utero, ASOs or whatever everybody wants to do. And in your case, you would have to give the mom and know early enough to give the mom minocycline, right? Like all this stuff is kind of crazy because when you're at a place like Baylor, I don't know if you've heard of it, like, those places have all the facility. So, they've got everybody getting RACGH, everybody getting whole exome, so that they do a different kind of science there than we do in Tennessee, for example. And so to imagine doing this kind of treatment is like just not there yet. You can't diagnose from fetal cells in the mom's blood because we don't even do that. Art might be doing that, but nobody else is doing that.
Yeah.
So nice thought.
Yeah, no, I agree. I think that it's challenging to start thinking about that from the human standpoint. One of the things that I'll be honest, we were trying a number of different things like the minocycline, and we're trying to think through some of those kind of synaptic modulation that might have might be helpful. But we know the problem with these mice. We know the problems with the humans. They don't have enough MEF2c. Now, we don't know exactly when, and we don't know if the effects of hypofunction are reversible. And so one of the things we have done is make a conditional rescue mouse. So we have a lock, stop, locks cassette that is basically, it's a knockout first, right? And so we can do that we can control the timing of when we turn it back on. Because the other problem, and this, right? This from Rett syndrome and MECP2 studies, this probably from Angelman and others, that too much or too little can be bad, right?
Yep.
And so if you're trying to think about a gene therapy, you can't just go in and start cranking out chicken beta actin expressed protein, or you're going to probably cause more problems than you had with 35% reduction in expression. And so we're trying a couple different things. One of the things, we're going to try this genetic rescue, because I think that's a proof of principle. Obviously, we're not going to do that in a human, but well, we might someday, but we're not going to be doing that in a human. But it's a proof of principle. Can we turn MEF2c back on at P10, at P28, in a young adult, and will it reverse some things?
I urge you to look at Epi Elgersma's paper if you haven't seen that on Angelman syndrome. Epi Elgersma did this with UB3A for his Angelman model, and he did all the readouts later on mice that had different reactivation of UB3A at different time points to show when he had to reactivate it, in order for them to not have these phenotypes anymore, like rotarod and stuff like that.
Right.
It's a really, really controlled paper. It's very, you'd like it. It's very much what you just said, but
Yep.
for UV3.
And I think it's important to think about, because, you know, we're now having a really greater appreciation of critical periods in development, right? That you know, microglia are playing a role in that, and we also MEF2c and microglia may be playing a role at a, you know, critical, precise periods of time when sensory integration is coding which synapses are kept and which are strengthened and so forth and so on. And so I think by having those kind of genetic tools, it's allowing us to be able to ask that question: when is too late? And also, because it's a flox allele, we can also ask it in cell type dependent manner, which is kind of cool. The other thing we're doing that's related to that is also along the lines of what several groups are thinking about for Fragile X and for Rett syndrome and a number, is also thinking about sort of the viral genetic therapy strategy.
Mm-hmm.
So if we know the cell types that are important, we have a number of you know, viruses that express MEF2c, and we can control which cell types and when they express it using a number of different tricks that you all know. You know, can we restore function? Because that is something we could do in a human. Now, humans, in principle, these kids start showing having seizures by, you know, between 12 and 24 months.
Yeah.
So if there is a window of reversibility, and if, you know, kids could get to that genotype, you know, that exome sequencing piece quickly,
Yeah.
there might be a window for intervention.
No, I'm not saying there isn't. We just haven't gotten there yet. That's all.
Yeah, and I don't know if this is going to be embryonic. I mean, my gut tells me a lot of the action of MEF2c is happening during embryonic development, but the fact that it plays this ongoing role in this sort of synaptic changes that are happening and that continues to be expressed in the adult brain, I don't think it's just about differentiation of neurons and then it's done. I think it's well situated to be able to play an ongoing role in remodeling.